In this lesson, we'll look at the cell cycle, a way of understanding the main events in the life of a cell. One of those main events is simply surviving, and we know that for a cell to survive, the nucleus of the cell must be making messenger RNA molecules, which are the temporary recipes to build proteins. That's important because proteins are the workers of the cell. They do all of the important jobs, from chemistry to building things to responding to the environment, etc. In addition, another important event in the life of a cell is reproduction. And for a cell to reproduce, it must duplicate its nucleus. Now, when we say duplicating the nucleus, after all, a nucleus is really just a membrane boundary in which chromosomes reside. So when we say a nucleus must be duplicated, it's really the chromosomes that must be duplicated. Those are the hereditary molecules composed of DNA. These are the recipes to make the proteins, the permanent recipes. But if one cell is going to make two, it must duplicate its chromosomes and thereby give its daughter cells a full set of chromosomes. And then each daughter cell will build a membrane around the chromosomes, forming its own nucleus. Here we see a single-celled paramecium, and if it's going to reproduce, it must duplicate its nucleus, making sure that each daughter cell will have a full set of chromosomes. On the right, we see paramecia stained red. In fact, the nucleus has the darker stain, and the two at the bottom here are caught in the act of dividing. Notice we have a duplicated nucleus here. The cell will split down the middle here, forming two cells. Each cell will have its nucleus. So the nucleus must be duplicated so that cell division results in two daughter cells, each with its own nucleus. We can think about the cell cycle using this circular graph. As we go around the circle, we're moving through time, and for the everyday person, there are three events that sort of define the life of a cell. Every cell has a birth date, right? So it's born from a pre-existing cell. The one exception would be Luca, born from chemistry, chemical evolution in those rocky chambers. But here we go. So a cell is born, and then it will go through a growth phase, and if it's lucky, if it survives and escapes predators, etc., it might get to reproduce. So we have birth, growth, reproduction, birth, growth, reproduction. And that's what's going on out there in the world. If we put our little paramecium into the mix here, we see a little paramecium here. It's going to be growing, it's eating, getting bigger. And remember, cells must remain small because they have a surface area problem as they get bigger. This applies to all cells, not just bacteria. So it's going to grow, 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 and then it will prepare for reproduction. So birth, growth, reproduction. One paramecium will be making two, and each of those paramecia will be going round and around the cell cycle. Now, biologists have a slightly different way of looking at the cell cycle. They've broken it up into two stages. One's called mitosis, the blue wedge here, and the other's called interphase. Now, if we think as time elapsing as we go around the circle, the larger area of the red part here, the interphase stage, what that means is interphase is a much longer stage of the cell cycle than mitosis, the smaller blue wedge here. So as we go around the cell cycle, the cell will be growing. At some point, it's going to have to duplicate its chromosomes. This occurs during interphase. Then the cell will prepare to deliver those duplicated chromosomes into daughter cells. Now, that process is the mitosis process. Some call it the nuclear division. It's the stage in which those duplicated chromosomes will be delivered to the daughter cells, and then each daughter cell will reassemble a membrane around those chromosomes, forming its own nucleus. Now let's take a closer look at what's going on during interphase. Well, here we said that the cell was sort of growing. Well, what does that mean to be growing? Well, metabolism is happening inside the cell, complex chemistry that involves extracting energy from the environment. The cell might be eating something, it might be just absorbing nutrients from the environment, it might be doing photosynthesis. So there's complex chemistry going on inside the cell. In addition, the cell is making proteins. Proteins are the workers. To do all that metabolism, you need a whole team of proteins doing different kinds of chemical reactions. And remember, proteins are produced from the temporary recipe of messenger RNA, which is a copy of DNA in the nucleus. Cells are also recycling proteins, they're repairing damaged proteins, etc. So if you're a paramecium, this is sort of what it looks like to be just living. You're eating bacteria, digesting, absorbing the nutrients, burning glucose, building proteins. If you're an amoeba, you're extending pseudopods with microtubules and choppers and receptor proteins. You're engulfing prey, and again, you're going to be digesting and absorbing nutrients and burning sugar, etc. So interphase, the first sort of stage of interphase is a growth period. Now, at some point then, if the cell is going to go on to reproduce, it's going to have to duplicate its chromosomes. Let's take a look at what that actually is. We'll go back to this diagram, and you'll recall that on the right here we have the DNA molecule, and then sort of as you go left, what's happening is the DNA is shown to be wrapping around these packaging proteins called histones, and sort of coiling up, and that's what we call a chromosome. But for a chromosome to be duplicated, that duplication happens precisely when the DNA molecule is duplicated. It's DNA that has the capacity for duplication. So chromosome duplication is really just the DNA molecule being duplicated, and each DNA molecule would then wrap up around proteins, forming 2 chromosomes that are attached still. Now that's important. So when DNA gets duplicated, the DNA molecule doesn't fully get duplicated. There's a region that is not fully duplicated so that these duplicated chromosomes can still be attached here. And they will not separate until mitosis. In the final part of interphase, after the chromosomes have been duplicated, the cell will get ready to do the actual division, to do the actual mitosis process. And in this stage, then, the cell is going to be making proteins that are required for mitosis. One of those proteins is none other than the microtubules. So we'll see that microtubules, those rod-like proteins, are going to be instrumental in handling the duplicated chromosomes, helping the cell deliver duplicated chromosomes to the daughter cells. Now let's take a brief look at what happens during mitosis. Well, the basic idea is that mitosis will be delivering a complete set of chromosomes to the daughter cells. And some people call this the nuclear division event. But now, what are these chromosomes? Well, actually, the word chromosome means colored body. And this word came into usage back in the 1800s when scientists were using various chemicals and stains to see if they could better visualize cells and cell parts. And what you're seeing here is a thin slice through an onion root tip. So the root of the onion plant, which of course grows down into the soil, if you cut a thin slice through that root tip and you stain it with a blue stain, this is what you would see. All the little rectangular boxes are root cells. When a root grows, it grows because more and more cells are being produced. That's what makes the root grow. Now, we're going to use this, we'll see a larger picture of this. We're going to use this to understand what mitosis is. Here we see a larger version of this, and you can start to see a little bit more detail now. In fact, most of the cells that you see here have a nice oval nucleus. It's as if the stain is nice and evenly staining the membrane of the nucleus. Those cells where you see an evenly stained nucleus, those cells are in interphase. And it makes sense that most of the cells are in interphase because, remember, interphase is the longest stage of the cell cycle. So you see, when they prepared this slice, they just kind of caught whatever cell was in whatever stage of the cell cycle. It was stopped at that point, and most cells would have been in some stage of interphase, and so we see most cells have even staining on the nucleus. However, there were some cells where you saw these little squiggles, and so scientists coined the term chromosome, meaning colored bodies, right? So these colored bodies are the chromosomes. And the suggestion was that sometimes you didn't see a nuclear membrane. Instead of seeing a nice stained nuclear membrane, you see these colored bodies. So perhaps these colored bodies were inside the nucleus. Well, what are these colored bodies doing? Well, if you kind of look carefully at the different arrangements of these colored bodies, you start to see a pattern. So, for example, here's a cell where the colored bodies are starting to become visible. Here's a cell where the colored bodies appear to be lined up. Here's a cell where the colored bodies appear to be sort of separated on either side of the cell. And here's a cell where the colored bodies now appear to be really darkly stained, and they are far apart in the cell. So what did all of this mean? Well, researchers figured out that what was going on here is that all those pictures of the colored bodies in different arrangements were really a stage, a series of stages in this process that we're now going to call mitosis. So when you start to see the little clumps of colored bodies, that was the first stage of this process of mitosis. Now, why would the stain start to clump up instead of a nice even staining nucleus? Well, the clumping would have been due to the DNA molecules coiling up and supercoiling, and that supercoiling would have allowed the stain to be more concentrated in these regions, and so we start to see the clumping of the stain. Now, what about the line-up stage here? Well, this would be the duplicated chromosomes being lined up at the center of the cell. Oh, it's starting to make sense now. Maybe what the cell is doing is it's going to be separating its chromosomes, and sure enough, that would make sense of this type of picture here, where the duplicated chromosomes are now moving to opposite sides of the cell. So one of these chromosomes is going to move to one side of the cell, the other chromosome will move to the other. So at this point, the chromosomes that were previously attached are going to detach so they can move to opposite sides of the cell. And then why would the chromosomes appear to be really far apart? Maybe because the cell's about to divide. So we're going to identify 4 stages of mitosis. Now, these are not the technical names for them, but we're going to name them with these names because they sort of describe what's happening in the stage. So condensing stage will be the first stage of mitosis, where the chromosomes are condensing and becoming visible. Line-up stage is the next, where the duplicated chromosomes are lined up. Separation stage, the duplicated chromosomes are separated, and division stage, the cell will form 2 new cells. Notice each daughter cell then will have a complete set of chromosomes. Now let's relate our picture here that shows sort of the structure of a chromosome to the process of mitosis. We can think then that what mitosis is doing here is taking those duplicated chromosomes and delivering them to daughter cells. In a sense then, the cell is moving the information in DNA into 2 daughter cells. So this is why the DNA is all coiled up and wrapped up, because the mitosis process, it's a moving process. You're gonna wrap up, you're gonna box up your stuff, and you're gonna move it somewhere else. So the chromosomes get all coiled up and compacted, so the cell has an easier time moving these things around. By contrast, during interphase of the cell cycle, the DNA is going to unwrap from those histones so that the DNA information is exposed and accessible, because now you need to access this information to make proteins. So this is our picture down here of the paramecium. Here we see the accessible DNA, and it can be used now to make messenger RNA molecules, which are temporary recipes, right? So during interphase, the cell is using the information in DNA to make proteins. During mitosis, the cell is moving the DNA information into 2 daughter cells. So using the information during interphase, moving the information during mitosis. And so we have a final summary diagram here of the cell cycle. We have interphase, the longest stage of the cell cycle, which includes a growth phase where metabolism and protein production and repair is going on. And also during interphase, chromosome duplication is occurring. Also during interphase, the cell will prepare itself for mitosis. Then mitosis itself has 4 stages. Those stages involve the chromosomes condensing, so they get compacted and coiled up, and then the chromosomes will be lined up, and then the chromosomes separated, and then 2 cells will form. So we'll call that a nuclear division event, and that's mitosis.